talk with you here. And the title of my presentation is EMP16 is required for mitochondrial AD2 intron for seed splicing and seed development image. The seed development from double fertilization, when the pollen tube reaches the ovary, they release two exactly same sperm. One sperm fertilizes an egg to develop into the embryo, and another sperm fertilizes two central nucleus to become an endosperm. After a period of development and combined with the pericarp, the maize kernel is matured that we can see directly. It's a kernel. And during the seed development, lots of organelles such as plastid, mitochondria, take part in. Today I will talk about the mitochondria. So the mitochondria play as the powerhouse of the cell. It provides energy for the cell division and differentiation. The plant mitochondria remains its own genome in the evolution. It can encode the proteins, RNAs, and tRNAs and RNAs. For example, the maize mitochondrial genome can encode the 34 proteins and 3 ribosome RNAs and 21 tRNAs. Although the maize mitochondria has its own genome, but the expression of the maize mitochondria under the regulation of the nucleus. The nucleus can encode the proteins transported to the mitochondria to govern the expression of the gene. One of them is the post-transcription governor. It includes the RNA editing, intron splicing, trimming, and RNA degradation. Today I will focus on the intron splicing. So the RNA splicing means different exons join together after intron spliced under the help of the splicing factor. And it has two types of splicing. Cis splicing means exons from the same pre-mRNA chain and the intron between them spliced under the help of the splicing factor, and then the exons join together. And the trans splicing means exons from the different pre-mRNA chain, and the intron between them recognize each other and form the secondary structure, and the help of the splicing factor, then the exons join together. There are lots of reports that the nuclear encoded splicing factors play functions in the whole splicing process. One of them is PPR proteins. So what are PPR proteins? PPR is a sort of the pentatricopeptide repeat protein, which consist of degenerated 35 amino acid motif repeated in tandem up to 30 times. In vivo, the PPR protein structure is similar and uses this region to bind the RNA. And study shows the PPR proteins mainly exist in the land plant, such as Arabidopsis and rest, but there are few PPR proteins in other organisms, such as animals and yeast. That means the PPR proteins are really important for the land plant. So the plant PPR proteins can be divided into two subfamilies based on the motif. The 25 amino acid motif called the P-type motif. Longer than 35 amino acid is a longer motif. Shorter than the 35 amino acid is a short motif. And the P subfamily PPR proteins consist of only by the P-type motif. And the PLL subfamily PPR are consisted by this motif. And furthermore, the PPR proteins in the PLL subfamily can divide it into 4 subgroups based on the C-terminal domain. The PLS subgroup and the E subgroup, E plus subgroup, and DYW subgroups. So the PPR proteins are encoded in nucleus and then transported to area corpus or mitochondria to perform the function. Its function are RNA editing, RNA processing, RNA splicing, and translation. So this is a short summary about the introduction that PPR proteins take part in the seed development through play functions in the mitochondria. Also, they can play functions in the corpus to impact the seed development, but I didn't mention in this slide. So when we talk about the maize seed development, we start from the development mutant. There are 3 types of seed development mutants. Empty paracap means both the embryo and endosperm are abnormal. So after desiccation, there are only paracap left on the air. And the embryo defective means just the embryo is defective, but the endosperm can well develop. So the seed is just smaller than the wild type and have the dark pod on the top of the kernel. And the defective kernel means the whole kernel, including the paracap, are abnormal. So the mutant involved in my project is the EMP phenotype. This is a phenotype of the EMP16. Around the 14 days after pollination, the pale homozygous mutant can be recognized. And after desiccation, it contains the traditional EMP phenotype. This is a kernel size comparison between wild type and mutant. In the cytological analysis from the 14 DAP to the 18 DAP, the embryo and the endosperm keeping growth in the wild type, but the development of the mutant is retarded. Also, there is a gap between the endosperm and the paracap, means the endosperm is retarded. So the basal endosperm transfer layer is essential for the nutrition transport from the material to the seed. So we use the specific antibody to test these cell layers. In the wild type, there are at least four layers of this cell, but in the mutant, there are only one, and the abnormal layer in the mutant. So after that, we want to know why. Our mutant library was built by mu transposon, which inserted in the maize genome randomly. So we use the mu flanking sequence to figure out the causative gene. The sequence result, based on the sequence result, we design a primer on the gene and combined with the mu transposon primer and perform the analysis, linkage analysis, and the segregation error also. All segregation errors have the insertion, and the non-segregation have no insertion, which means the linkage is tight, and also we isolate another allele from the library, and the cross between this. The cross between these two alleles also contains the EMP phenotype. So we confirm this gene is a causative gene for the phenotype. Also, this allele with the mu transposon inserted in the file UTR, but there is no phenotype. It is common because the mu transposon inserted in the file UTR will not knock down the gene. So after clonal gene, this gene encoded a PPR protein with 11 motifs. The alignment of these motifs shows highly conserved. So this is an expression of this gene in the 3 different alleles. Also, this PPR protein is universal expression in all organelles, not just kernel specific. It consists with the reported PPR proteins. So before we want to know the function of this protein, we perform the subcellular localization of this protein. The GFP signal is merged with the mitochondria, which is the specific staining of the mitochondria, means this protein localized in the mitochondria. So the mitochondria just have the 34 protein coding genes. So we use the RT-PCR to screen all these genes. The result shows that the NAD2 has dramatically decreased in the mutant. And the NAD2 is the cis splicing gene. They have two, NAD2 is a trans splicing gene. They have two trans splicing introns and two cis splicing introns. We design primers to cover this region and detect the splicing efficiency respectively. And when we use this pair primer, we can't amplify the normal fragment. And this fragment contains the unspliced intron and was confirmed by the sequence result. Also, there's a report the editing and the splicing are involved together. So we test all the editing side of the NAD2 and they are all normal. That means the splicing and editing, RNA splicing and editing are independent. Also, the NAD2 is a member of the complex 1 in the respiration chain. So the unspliced NAD2 can't encode a normal NAD2 protein. That will influence the assembly of the complex 1. Then we used the beam page gel to test the complex 1. And the result shows that the complex 1 is decreased in the mutant. Also in the in-gel activity assay, the activity of the complex 1 is consistent with the Comas blue stain. So in the respiration chain, the defective complex 1 will stimulate the succinate pathway that will induce the expression of LX genes. So we test the LX genes using a real-time PCR, and the result shows the LX2 and LX3 is highly induced in the mutant. This report combined with the blue native gel supported that the defective of the NAD2 intron for splicing. So this is a take-home message. EMP16 encoded a PPR protein that functions on NAD2 intron for cis splicing in maize mitochondria. Abnormal NAD2 leads to the defective of complex 1 in mitochondria. Such defective blocks the development of maize seed embryo and endosperm. Splicing and editing are two independent processes, at least for the NAD2. Finally, I give my acknowledgement to my supervisor, Professor Tan, and Professor Zhang, and also Dr. Nakagawa in Japan to provide the vector. All lab mates in my lab, and this project was supported by the National Nature Science Foundation of China. Thank you. Questions are welcome.
Any questions for?
Yes, I have.
Sure, go ahead.
Thank you for your nice presentation. I have one question because I didn't understand this probably. So you have used mutants by using transposon tagging.
Yeah.
So when you tagged, so you went after the phenotype of the either knockdown or knockout.
It's a knockout.
Knockout.
Yeah.
So, but in that case, how we clone the gene? How did you? What is the method you use?
It's a sequence technique. We use when the transposon inserted in the gene left a flanking sequence, we can use the transposon-specific primer to sequence the flanking sequence.
How can you go to the genomic DNA? Because this is your transposon, this is your sequence, right?
Yeah.
If you do primer, if you do PCR, you always amplify this internal fragment, so-called transposon fragment. How do you come to the genomic fragment? Because this genomic fragment is unknown.
Well, we use the adapter primer that with I don't know how to the specific the correct we use adapter like an adapter primer and use the transposon specific primer to amplify, and when we run the DNGel, we can compare a different band between the wild type and the mutant. But in the mutant, but in the in-gel picture, the mutant will have this fragment, but the wild type will have no, because this mu transposon didn't insert it in the related gene. We use the adapter primer and the mu transposon specific primer to run the PCR. Then we separate.
It's a classical way of doing
Yeah.
gene pull out with transposition, especially with the mu transposition. It's more of more of a universal adapter sequence, which you prime it at low conditions and then use higher stringency to pull out your specific band.
I call it the mutile PCR. Yeah, mutile.
Yeah.
Just you do not use this chromosome walking or RFP or inverse PCR.
Yeah.
It's the same thing what people use for flanking sequence analysis. It's like more of a
It's similar with TDN insertion in the
Yeah.
arabidopsis.
Yes,
Yeah.
but in that case, people are using this inverse PCR. Once they
Yeah.
record this and circulate these things, and you make one PCR using the internal sequence and the transposal sequence as a prime. So you have to lag it, make the circle. That's why I didn't understand how you express transposon, because if you use the transposal sequences as a primer, you always end up with the same prime.
Yeah.
So you have no
one is
idea.
transposon specific and one is more of a
Yeah.
universal adapter. We can discuss more of the details later. Anybody else has a question? Okay, I'm going to ask you a question.
Yeah.
How are you going to use your understanding to make improvements in seed development, maize seed development? The NAD2, for example. How do you use this knowledge?
You mean
How are you going to find your discovery in application? How are you going to use it for?
I think it will take a long time because it's just the basic basic research, and we just want to find the related gene for the seed development, and maybe the company or the other research focused on the applied will use our finding. Yeah.
Any more questions?
Just one question. I wonder if there is any alternative splicing occurred in this gene. Alternative splicing, intron alternative splicing.
No, there is no intron. It's a gene without intron, the EMP16. Yeah, just with the mu transposon inserted in the mutant.